This is your Honors Biology Semester 1 review. Please take note of your test dates. First question asks us about biased data. Now, biased data is any information in our experiment that is going to unfairly favor one variable or another. And it's usually due to a flaw in the experimental design. So an example, let's say that you're a group of doctors and you are trying to test out a new drug, and this drug is supposed to stimulate hair growth. If you tell your patients this, all of a sudden the patients start reporting hair growth. Now, this is more than likely due to the fact that they are expecting hair growth. So because they are expecting that result, they'll favor it, even if the drug is just a sugar pill. So this is why whenever they do a drug test, they do a double-blind experiment. The scientists don't know, and the patient doesn't necessarily know anything about what drug they are taking, so that there is no biased result. Next question is about the cell membrane. So a few important things to point out here. It is semi-permeable, which means that some things can pass through and others cannot. The cell can pick, and anything that is entering or exiting the cell must go through this cell membrane. It may go through a transmembrane protein. So that could be something like an aquaporin, which allows water through, but it is a transmembrane protein that allows substances to travel through, into, and out of the cell. Other important things in here, this carbohydrate side chain, that is important for cell-to-cell recognition. If cells are in contact with each other, those carbohydrate side chains are recognized by the glycoproteins, and that allows the cells to know that they are amongst friends, that they are in fact next to the same type of cell. Other thing I want to point out is the phospholipid. So the phospholipid is the building block of the cell membrane. A phospholipid has this hydrophilic head, that means water-loving, and these hydrophobic tails, which means water-hating. So what actually happens if you were to drop a bunch of these guys into water, they would self-assemble, they would arrange themselves so that these hydrophobic parts are in the middle, kind of sandwiched in between like we see here. And the hydrophilic parts would be on the outside facing the water, like we see here. This is called the fluid mosaic membrane model because all of these proteins and these things that are sticking out of the membrane, they are able to move around within the membrane. The lipid that is found in all cell membranes is phospholipids. The polar head is the phosphate group, and the nonpolar tail is hydrocarbon. So polar, that's what water is, so that's where the hydrophilic part comes from, water-loving, and nonpolar is something like oil, so you know that oil and water don't mix, so that is why it is hydrophobic. They form a bilayer, which makes up the cell membrane. So, small, polar, uncharged molecules, such as water, can slide through the cell membrane. That's also carbon dioxide. The polar ends of the phospholipids attract the polar molecules. Most cells also have aquaporins, so that's a specialized protein, a specialized water channel to help get water through. Next question asks us about scientific notation. So here's a couple examples. So to write 1,000 in scientific notation, you take your decimal point, and that's normally at the end, and we count over 1, 2, 3 spaces. So this is 1 times 10 to the positive 3. The reason it's positive is because we had to move the decimal place to the left, which means that this was a really big number. So 1 times 10 to the 3 is the same thing as 1,000. This number right here, I'm going to take my decimal place and I'm going to move it over 1, 2, 3, 4, 5. So I got 2.35 times 10 to the negative 5th. And the reason it's negative, we move the decimal place that way towards the right, which means that it must be a very small number. The decimal place gets moved over until you have just one non-zero number to the left of it, so we moved it over till we just had the 2 to the left of the decimal place. So this question says to convert 3 micrometers into millimeters. So 3 micrometers, and this is the symbol, it looks kind of like a U, that means micrometers, and I want to figure out how many millimeters is that. So the difference between micrometer and millimeter is a factor of 1,000, which is a 10 to the third difference. And micrometers are smaller than a millimeter. So 3 micrometers is going to be 3 times 10 to the negative 3 millimeters. And this is because the difference between them is a factor of 1,000, so 10 to the third, and a micrometer is 1,000 times smaller, so we've got the negative 3 because it's smaller. So difference between a control and a controlled variable. So a control in an experiment is a point of comparison. It's a trial in which the independent variable cannot possibly affect the outcome. So if I'm testing the effect of color of light on photosynthesis rate, the control would be a plant grown in normal light, and my experimental groups would be a plant grown in red light, or blue light, green light, whatever other color lights I want to test. A controlled variable is something that can change in an experiment, so scientists have to account for that. So for example, the plants might encounter a range of temperature. Ideally, I would have my temperature to always be constant, and I would pick, room temperature, 72 degrees Celsius, or something like that. I would pick a temperature, 72 degrees Fahrenheit rather, and that would be constant. But I can't always do that in an experiment. So to account for this, I would make sure that all of my plants experience the same range of temperatures. So if my plants are in a room where the temperature varies by 15 degrees Fahrenheit, I would want to make sure that all of my plants encountered that same temperature range. Otherwise, I wouldn't know that the only reason plant A grew faster was because of the color of light. It might be because plant A was colder than the other plants, so it didn't photosynthesize as quickly. Enzymes are very important. They are biological molecules, and they speed up the rate of chemical reactions. A lot of the chemical reactions necessary for life are very, very slow, so slow that they possibly couldn't support life. So in order to speed up these reactions so that they occur at a rate fast enough, you wouldn't want to be waiting around for 5 days to get the energy from the hamburger that you ate. So we have enzymes that speed up these reactions so that they're more usable and more reasonable for life. So examples, catalase, which is what breaks down hydrogen peroxide in cells. So that's a byproduct of hydrogen peroxide, and it's toxic, so it needs to be broken down, and that's done by catalase. Proteinase, that would be something that would break down a protein. And lactase, of course, breaks down lactose. And the things that these all have in common is that they end with ase. So anytime that you see a molecule that ends with an ase, it is more than likely an enzyme. They're made of a bunch of proteins that have been folded and twisted together in a complex three-dimensional shape. That particular complex shape is what makes enzymes so specific. So catalase only does a specific function. Lactase only works on lactose, and that's because of this complex three-dimensional shape. So here's kind of a model of what this looks like. It's the exact right shape to fit lactase, to fit lactose. So lactase breaks down lactose, which is milk sugar. If you're lactose intolerant, you do not have enough of this protein, and that could be due to several factors, but one way or another you don't have enough lactase. So people will take medicine from, you can get it over the counter, and they will take that because it's got extra lactase in it, so that if they do eat something with milk in it, they're able to break down that lactose. Carbohydrates, the building blocks would be a monosaccharide. So here we have glucose, a monosaccharide, and fructose, another monosaccharide. Whenever you put those two together, you get a sucrose, which is a simple sugar, simple carbohydrate. The building block for nucleic acids are nucleotides. So the nucleotides are A, T, G, and C for DNA. We also have another nucleotide for RNA, but the main four are ATGC for DNA and AUGC for RNA. So nucleotides is what nucleic acids are made of, and that includes DNA and RNA. DNA is for deoxyribonucleic acid, and RNA is for ribonucleic acid. And the building block for proteins is amino acids. So there are dozens and dozens of different types of amino acids, and depending on what order they are linked together in, we get the different types of proteins. This R group right here, we call this an R group. This can be a different molecule depending on what the amino acid is. So this part right here, this is the same for all amino acids. This part changes for each type of amino acid. Carbohydrates are used in humans as the main energy source. This is why our diet should be mostly carbohydrates. An example is glucose. Notice that that ends with ose. That makes it a sugar. It is used by cells in cellular respiration to make up ATP, which is a usable form of cellular energy. Glucose is often called blood sugar. In plants, carbohydrates may be found as a structural purpose. So cellulose, again, it ends with ose, so we know that that's a sugar. And cellulose is what the plant is made of. So, for example, paper, which is made from trees, that's just cellulose. Some other animals use more complex carbohydrates, such as starches, like chitin, which is the exoskeleton of horseshoe crabs, most insects. They have this exoskeleton, which is actually made from very complex carbohydrates called starches, and chitin is an example of that. The vitamins. Vitamin C is important for wound healing. Vitamin D is for bone formation, and vitamin K is for blood clotting. So vitamins are sometimes called coenzymes, and that's because they work together with enzymes to help them perform their assigned task. So vitamin C doesn't just heal wounds. You can't dump vitamin C on a cut and it closes up. It would help the structures that are already existent within your body to heal that wound. Differences between plants and animal cells. So, big difference is that a plant cell has a rigid cell wall, which is again made of cellulose, a structural carbohydrate. And that cell wall is rigid, so it holds that plant into a very rigid straight shape. A vacuole. So inside of an animal cell, you have small little vacuoles. In a plant cell, you have a big vacuole. This is kind of like a storage facility for the cell. Animal cells can store wastes and that kind of thing in the vacuole. Usually in animal cells, that would be broken down by some enzyme. So hydrogen peroxide might be stored in a vacuole in a plant cell. In an animal cell, and then broken down by an enzyme. In plant cells, there's usually just the one big huge one, and that's usually for storing water. Sometimes they can have sap, sugar, salts, that kind of thing in there. The other big thing that makes plants really special is that they have chloroplasts. And this is important for photosynthesis. There are other pigments besides chloroplasts that are important for photosynthesis, but chloroplasts are the main one that we worry about, and this is what gives plants their green color. They both have mitochondria, so that is essential for cellular respiration. They both have that, so that's how they get their energy. The cell membrane is present on both. And it's used to control what goes in and out of the cell. They both, of course, have cytoplasm, and they both, of course, have a nucleus. Now, generally speaking, plant and animal cells are very different, and you can just look at a microscope for that cell wall, but there are some exceptions, and there are actually a few photosynthetic animals that exist as well too, which are very interesting to learn about. And you can, of course, find those on YouTube. So, diffusion. Diffusion is the movement of particles from a high to low concentration. So you see in this picture, I've got these particles, and they are at high concentration on this side, and the net flow of them is going to be in this direction because there are no particles over here, low concentration of particles. It is due to a concentration gradient, so that's what we see here. We see that we have one side with high concentration and one side with low concentration. So that difference is what we call the concentration gradient. This occurs from random collisions. So these particles bump into each other, and that causes them to kind of diffuse apart. A higher temperature means that the particles are moving faster, so that would be more collisions, and that of course means faster diffusion. Next question is asking us about the difference between osmosis and diffusion. So diffusion, as we saw on the last slide, is just random spreading out of particles, and it's just due to their collisions. Osmosis is a special case of diffusion. This is where the thing that is diffusing is water, and it is diffusing across a semi-permeable membrane. So the difference is diffusion could be anything. Osmosis is specifically water across a semi-permeable membrane. So here we've got a picture, and in this picture I've got two tanks of water, and they are separated here by a semi-permeable membrane. Okay, so that's what that is. That's the semi-permeable membrane. On this side, we've got some sugar. So we've got 3 sugar molecules. And we see that the water level is higher on the side with the sugars than it is on the side without the sugars. The reason why, around each of these sugars, so that's my sugar, okay, the water molecules are bound to the outside of this. So all of my little water molecules are stuck to the outside of this. This is called a solvation shell. So the concentration, or the number of free water molecules, is less on the side with the sugar because a bunch of them are bound. So because the sugar side has less free water, in order to make the amount equal on both sides, water would flow this way so that there's the same amount of free water molecules on both sides of the cell membrane. So if I have a cell that lives in 20% water, salt water, then the salt concentration inside of that cell would be 20%. So if I were to take that cell and I were to place it into pure water, so that would be a situation like this. So there's my beaker, here's my cell, and this cell is 20%, and pure water would be 0% salt. Because the concentration of salt inside of the cell is higher, there's fewer free water molecules, so all of the water is going to go into the cell. This causes the cell to burst. We call that situation hypotonic. The concentration of water outside of the cell is less than the concentration of water on the inside of the cell. So, in another situation, I've got my same cell that is 20% salt, and I drop it into really salty 80% water. So in this case, the concentration of solute is less inside of the cell, so water is going to flow out of the cell, and this causes the cell to shrivel and shrink, and we call that hypotonic. Hypertonic, rather. Hyper means above, so hypertonic would be something that is higher concentration outside than inside. So in plant cells, they have that rigid cell wall made of cellulose, which prevents the cell from bursting if it was placed into a hypotonic solution. So if you took that cell, it would have something that prevented that cell from taking on any more water, and that's that cell wall. So here we see in a plant cell, if it is hypertonic, that means the outside is more concentrated than inside. All the water flows out of the cell, and we see the cell membrane here has shrank. In isotonic, that means that the concentrations are equal, so just as much water flows in as flows out. In hypotonic, that would mean that the inside of the cell is more concentrated with solutes than the outside of the cell. Water would flow into the cell, but this cell wall here is preventing this cell from really expanding to the point of bursting. And you see that the water vacuole gets bigger too. So the difference between acidic and basic, anything that is pH 7 would be considered neutral. Below 7 is acidic, and above 7 is basic. So right around basic, you see that you have seawater is going to be slightly basic, whereas water from the sky, rain, would be slightly acidic, and that is due to the different chemicals that are present in our atmosphere. Stream water, fresh water, is normally right around neutral. Enzymes. So here we show a couple different proteases. So there's one found gastric and there's one found in the intestine. So one in the stomach, one in the intestine. And you see that the relative rate of activity increases, has a maxima around a certain pH. So this pH right here, so the intestinal protease works best at a pH of 9. The gastric protease works best around 3. And we know this because we have the highest rate of activity at those pHs. In this instance, the independent variable would be the pH, so that goes along the x-axis, and the dependent variable would be enzyme activity along the y-axis. Here's another enzyme, and we see that this enzyme right here has a maxima, so it has the most activity right around 28 degrees Celsius. So for this one, the independent variable is temperature on the y-axis. We have enzyme activity, which is the dependent variable. Temperature is independent, which is on the x-axis. So if you take an enzyme, and we saw in the previous slide that they work best within a certain range of temperatures and pHs. If you take an enzyme out of those optimum conditions, that can cause the enzyme to denature. So in this picture, in this graph, we see that right after around, right around here, we see that there's sudden decrease in enzyme activity. And this is because the enzyme has become denatured. It's lost its shape and it can no longer function. Here's the equation for photosynthesis. So what plants do, they take in carbon dioxide from the air, water from the soil through the roots, and light strikes the chlorophyll. And that energy is used to create glucose, which is long-term energy storage, and oxygen is given off as a byproduct as well. Nothing can just use glucose. It needs to break the glucose down into ATP. So plants are the only things that photosynthesize, except for a few of the weirdos of the animal kingdom. And all organisms that use glucose for energy, they need to break it down through cellular respiration. Carbon dioxide is a required reactant. If you were to increase the amount of carbon dioxide available, you should speed up the rate of photosynthesis. If I decrease the amount of carbon dioxide present, there's less CO2 to make sugars with, so the reaction would slow. Sunlight is also a required reactant. So more sunlight would give me more solar energy to make sugars. Less sunlight would be less energy available to make sugars. Now the trick with this is everything is within a certain range. At some point, there's so much carbon dioxide that if you add more, it's not going to make a difference because the plants are photosynthesizing as fast as they are able to. So at that point, it would no longer be dependent on carbon dioxide. It would be dependent on some other variable. Same thing with sunlight. Too much sunlight can make things very dry, and that can actually make less water available to plants, and that can actually slow down the rate of photosynthesis and can harm the plant. Chloroplasts are where we find chlorophyll. Those are the pigments that absorb sunlight to use it to make sugars. So this is a chloroplast, and this would be inside of a plant cell. So this is not a plant cell. This is just an organelle within a plant cell. So you'd have a bunch of these, thousands of them within a plant cell, and you see that this organelle is broken up into even smaller structures, but we find that this is where the chlorophyll are found, and that is why this is green. So, a way that you could use a water plant. If you take a water plant, an Elidea, and you put it into a graduated cylinder, and you can turn it upside down, as that plant photosynthesizes, it creates oxygen. So if you leave that cylinder on top of it and you remove all the air, the only gas inside of that after an extended period of time would be oxygen. So you can figure out how much oxygen was collected over an hour or over a day or whatever, and use that to figure out the rate of photosynthesis. So here we've got my Elodea. It's a water plant, and there is a graduated cylinder. So these bubbles that are formed by these plants are being collected in this graduated cylinder, and I can measure the amount made over a certain period of time. So the first picture is an animal cell. It is round, so that means that it doesn't have a cell wall, and it's got a nucleus. It is taking in glucose, so in this picture, glucose goes into process A. Process A takes place inside this mitochondria, and it spits out ATP and CO2. So this is cellular respiration that's occurring. The second cell is a plant cell, and I know this because I've got a cell membrane here, or excuse me, a cell wall. And we see that CO2 goes into process B, and process B makes glucose. And this is taking place inside of a chloroplast. So that would be photosynthesis. The last image is showing some muscles on a human arm. So in this case, glucose is going into process C and producing lactic acid. Whenever there's no oxygen available for cellular respiration, the cells will instead go through fermentation, which is a type of anaerobic respiration. So that glucose will be broken down and it will yield lactic acid as a byproduct. Over time, that lactic acid can build up on your muscles, and that's what leads to cramps and that sort of thing from working out too much. So different types of cellular respiration: aerobic, which means with oxygen; anaerobic, which means without oxygen. So aerobic means with oxygen, and if you put an "and" in front of that, that means without oxygen. So, aerobic respiration and anaerobic respiration. The first step is glycolysis, which is just splitting apart sugar. That takes place in the cytoplasm. For aerobic respiration, the products of glycolysis then move into the mitochondria, where they'll undergo 2 more steps. So we'll have eventually electron chain, and we will break down that sugar further until what we get is a total of 38 ATP produced for 1 glucose molecule. And here's the equation. The big deal here is that we have this O2 here, and the energy that I'm talking about is ATP. In anaerobic respiration, after glycolysis, the broken parts of the sugar will enter into fermentation. We only get 2 ATP out of this, so this is much less efficient. And here you see that there's no oxygen. In this case, this is ethanol. So this would be alcoholic fermentation. You could also go lactic acid fermentation, which is what we saw previously in the last slide. So for anaerobic respiration in muscle cells, you would make lactic acid instead of that ethanol. Cellular energy. The molecule that is used for cellular energy is ATP. It is made up of adenosine, ribose, and phosphate. So ATP stands for adenosine triphosphate. So there's one adenine, one ribose, and 3 phosphates. So these wavy lines between the phosphates, those represent very high energy bonds. So whenever you make these bonds, you store energy. And if you were to cleave one of those bonds, if you were to break it off, so if you were to take the ATP, break it off, here you would make ADP, but you would also release a lot of energy. So that's how ATP is usable cell energy. We take that energy originally stored by plants in glucose from the sun, and we break that off to release that energy whenever the cell needs it. So aerobic and anaerobic. If a muscle runs out of oxygen, it can no longer go through aerobic respiration to make ATP. And this is like a split-second decision. 1 second, they might be undergoing aerobic respiration, and then a second later, they might be undergoing anaerobic, and then a split second later, they might switch back to aerobic. It just depends on how much oxygen is available in that instant. Anaerobic respiration, also known as fermentation, can keep a cell alive by making a small amount of ATP without oxygen. So if temporarily there's not enough oxygen for your cell to get energy, it can undergo fermentation, keep itself alive until oxygen becomes available again. You will know that anaerobic respiration has happened because muscles will cramp up, and that's because lactic acid has built up on them, which is a byproduct of fermentation. The carbon cycle. So photosynthesis produces sugar that respiration uses, and cellular respiration produces the carbon dioxide needed for photosynthesis. So here we've got a picture of a carbon cycle. So there's carbon dioxide in the atmosphere. It's also dissolved in water. Photosynthesis occurs in green plants, which produces sugars. These sugars can be used by animals through cellular respiration, and they produce more carbon dioxide. The other option is that whenever those plants die, or whenever those animals die, those carbohydrates and those sugars are decayed by bacteria, and then that releases the carbon dioxide back into the atmosphere. Alternatively, those plants and animals may decompose into the ground, and they may eventually become fossil fuels, which through combustion, such as in cars, for electricity, all of that, releases the CO2 back into the atmosphere, where it is then retaken up by plants, and the process can continue. Bromothymol blue, also known as BTB, is what we call an indicator. Indicators are used to detect the pH of a solution. Now, if we add carbon dioxide to water, we form carbonic acid, making the water more acidic and changing the pH. So, bromothymol blue changes from blue to yellow when carbon dioxide is added because we form that acidic solution. So if I were to have my 4 beakers here, beaker 1 has got nothing, beaker 2 is a plant, beaker 3 has a snail, beaker 4 has a plant and a snail, nothing will happen in beaker 1 because there's nothing, no processes going on. So if you add bromothymol blue, it will stay blue. Plants will remove any CO2 from the water. So because there's no extra CO2, it's all being taken out of the water. It would stay blue as well. In this case, the plant would eventually die because there's nothing to replace the CO2, so that would be bad news for the plant. In the snail beaker, cellular respiration is occurring, and that is adding carbon dioxide. So the water will change to yellow. And the snail will also die because there is nothing to take up its carbon dioxide and make sugars for it to have as food. In the fourth beaker, you've got both processes occurring. So cellular respiration is adding CO2, photosynthesis is removing, and removing the CO2. So depending on how you explain this, the water might stay blue, or it might turn green because both processes are in balance. What cells contain mitochondria? So, any cell that undergoes cellular respiration must have mitochondria. So therefore, all eukaryotes contain mitochondria, because this is a membrane-bound organelle. Prokaryotes do not have membrane-bound organelles like the mitochondria, and they rely on fermentation for ATP generation, which just occurs in the cytoplasm. So all eukaryotes, plant cells, animal cells, all eukaryotes have mitochondria, but prokaryotes do not, because they lack membrane-bound organelles. What is meant by cellular energy? So cellular energy is ATP, which is just a usable form of energy for the cell. Pure glucose is not something that the cell can use. It needs to take that energy and convert it into ATP whenever it needs energy. The organic molecule would be sugars, which are used in cellular respiration to make the ATP. So sugars are a long-term storage option for energy. The cell, the organism, plants store this, and they can use that whenever they need energy as ATP. ATP is a very short-lived, unstable molecule, so it doesn't hang around for very long. So organisms need to store sugar so they can make ATP whenever it's needed. Differences between prokaryotic and eukaryotic cells. So the big difference is that prokaryotic cells lack membrane-bound organelles and a nucleus, and eukaryotic cells do have those. All cells have ribosomes, which are necessary for protein synthesis. They have a few other functions, but that is necessary in all cells. And they all have cell membrane, because every cell needs to control what is entering and exiting. That's how it maintains homeostasis, how its internal environment is different from the external environment, and that's just by controlling what enters and exits the cell. Prokaryotic cells have a cell wall made of peptoglycan, whereas eukaryotic cells only have a cell wall in the case of plants, where it is made of cellulose. DNA for prokaryotic cells is very simple. It's just a circular loop of DNA, whereas in eukaryotic cells there is long linear strands of DNA that is found in the nucleus. Pro sounds like a professional, somebody's been doing something for a long time. So prokaryotic cells are old cells. They've been around for a long time. Eu sounds like new. So eukaryotic cells are more modern cells. Prokaryotic cells are considered primitive because they have no membrane-bound organelles. They have a single loop of DNA, which just floats around in that cytoplasm. And they've been around for a long time. The very first living cells on Earth were prokaryotic cells. Vascular tissue. So in plants and animals, this can include the circulatory system, but specifically in plants, we have the xylem and we have the phloem. So the xylem is a system of tubes that transports water and minerals, which comes from the soil, and it provides support. As the xylem dies, it forms the rings that you see whenever you look at a tree and you can count how many years it's been around. The phloem takes sugars and transports them in addition to other molecules that the plant needs. And the phloem is always alive. That's the green sapwood that you find in trees. So here's a xylem, and you see that in the xylem it is one way. We've got water and we've got minerals mixed in. There's no walls between the cells, and they are very thick and they are stiffened with ligands. So these are the structural support part of the tree. The phloem is kind of got these little components here, and it can have two-way flow. And we see that we have food, sugars, that sort of stuff being transported. So different types of cells have different strategies for movement. So here, this cell uses cilia for movement. So these cilia are these little tiny hair-like projections. They're like little oars that the cell can beat to move around. An amoeba uses pseudopoda, false feet. So it takes parts of its cell and kind of stretches it forward and pulls by dragging itself along. Last one, this is an organism that would use a flagellum to move. So that is a long whip-like structure. The cell can kind of flail that back and forth to provide movement. So the circulatory system circulates materials through the body, such as oxygen and hormones. So this would be, in your case, the blood. Once oxygen diffuses through a cell membrane, it can then enter the mitochondria, where aerobic respiration can begin to generate ATP. And ATP is used to do work, it's cellular energy around the cell. So the circulatory system does a whole host of things you may not necessarily think of the circulatory system as doing. So it transports body fluids by delivering water and nutrients to cells as blood. It can also fight infection. It will carry white blood cells, which can destroy bacteria and other foreign invaders in the body. It regulates body temperature. Whenever you start to get cold, your capillaries inside of your skin, they close up, and your body will pool that blood in the most important organs, in your chest, and the most important internal organs to help keep them warm. Whenever you get hot, your capillaries dissipate, and that blood carries that heat near the surface of your skin, where it can then transfer to sweat, or it can transfer to the air outside. The circulatory system also transport chemical messengers, such as hormones. This allows your body to react to its environment. So your body may react to protect itself. It might tell you that your blood sugar is low and make you feel hungry, or give you a fever, or tell your body to shiver to keep warm. Hormones are the chemical messengers transported by the circulatory system and made by the endocrine system. So the endocrine system is just a series of glands which make and secrete hormones. These hormones, these chemical messengers, can be sent very long distances by the circulatory system. And the precursor for hormones is cholesterol. So cholesterol is important in your diet so that all of these hormones can make. In particular, testosterone is one that's made from cholesterol. So here, there's a picture of endocrine system glands. So you see these glands are all over the place. You have pituitary gland, pineal gland, hypothalamus. These are all in your brain. Thyroid gland, parathyroid glands. Within your body, you have your pancreas, adrenal glands, ovaries, testes. These are all glands that produce hormones that are important for human function. So the nervous system regulates. It's one of the regulation systems because it senses stimuli, changes in the environment, and that nervous system can then send that signal, and the endocrine system may make chemical messengers to account for the change or to respond to the stimuli. So the nervous system might sense that the air outside is cold, and that might tell your brain that it needs to tell the body to shiver so you can warm up. This is important for homeostasis, which is the unique set of circumstances needed for a cell to live. So this is things like just the right temperature, just the right oxygen, just the right pH. These are the Goldilocks conditions in order for a cell to survive. And if we cannot maintain these conditions, the cell will die. So an example is the regulation of blood glucose. So inside of your body, you have glucose in your blood. And if it gets too high, the body releases insulin into the blood. Insulin tells the liver to take that glucose and store it as glycogen. So it tells the liver and muscle and fat tissue to take up the glucose and store it so that the level of glucose in the blood goes down. People who have diabetes don't have enough insulin. This is either due to an autoimmune disorder or due to lifestyle, so they're eating lots and lots of glucose, lots and lots of sugary foods, that kind of thing. And they will not be able to maintain homeostasis if they do not take medication. So if they don't take medication, they get a blood sugar spike, and that can lead to coma and eventually to death. Getting rid of waste is another important thing for cells to do to maintain homeostasis. And the main system that gets rid of waste is going to be the cell membrane. All things going into or out of the cell must pass through the cell membrane. That includes wastes. So, in different animals, there might be other systems in addition to the cell membrane that help to get rid of waste. But the first gateway for that waste to get through is the cell membrane. It might go through and turn to the blood and go through an excretory system or some other system to excrete it from the organism, but the first place that waste needs to pass through is that cell membrane. So it's often called the gateway to the cell because everything must go through it to enter or exit the cell. So in humans, here's an example of a stimulus. If you put your hand close to something that's hot, so that is the stimulus, you're going to say ouch, and your response to that is of course going to be to jump away or to pull your hand back. So a stimulus is something that leads to a response, and the purpose of this is to protect that organism. So how is a stimulus sensed? So in this picture, this is a reflex arc, which is part of the nervous system. So here we've got the skin, and these represent the nerves in the skin. So if I were to take a pin and poke your skin, these nerves would detect that. It then sends that signal through the axon, so that's what this thing represents here. The axon is made of myelin, where it will go through a cell body. So this kind of like transfers this from the sensory side of the axon, from the nerve side, and that will go to the motor side. So this is connected again by an axon made of myelin, and that is connected to a muscle cell. So in the last picture, whenever the hand touched the fire, that would be sensed by the sensory nerves within the skin. That impulse, that signal, would be carried to a cell body, which would then be carried to a muscle, and of course the response to that would be to jump away. Feedback mechanisms. These are important for maintaining homeostasis. It's the way that the body or an organism will respond to environmental changes, such as temperature. So in this picture, we see that without sweating and shivering, humans could not maintain their homeostatic body temperature. So let's say that it's cold. So it's too cold, right? The skin senses that, and what happens is it causes the blood vessels to constrict or prevents them from sweating. Eventually, that causes the body temperature, the blood temperature, to increase. The brain senses this and tells the skin, you're fine, you don't need to shiver anymore. Or if you're too hot, it tells that skin to sweat or to dilate, open up to get rid of that heat until body temperature decreases. So that signal is carried via blood and via nerves from skin to the brain, so that the brain can kind of help the body either shiver when necessary, sweat when necessary, or do neither if temperature is okay.